Okay, I think we can slowly make a start. Welcome to this webinar hosted by the Centre for Longitudinal Studies. It's on the topic of getting started, an introduction to 4 British cohort studies. Thank you very much for joining us. We're really excited to introduce you to these important data sets. My name is Morag Henderson, and I'm here with Richard Silverwood to introduce you to the value of the cohort studies for research, the sort of contents of the studies, and some of the things that you might want to do for your own analysis and getting started with the studies, how to access and where to go for more information. So a few housekeeping things before we start. Your cameras and mics are already turned off. That's just because we're recording this session, and it allows us to put the recording on YouTube channel for future reference, and we don't want too many interruptions during the actual presentation, but we would encourage you to put any questions that you have in the chat. Please note that your questions will be visible to all attendees, and we will store up those questions and answer those at the end of today's session. If you have any technical issues, please email us at ioe.clsevents@ucl.ac.uk. I mentioned that we're recording the session. We will also send you a link to that recording and slides afterwards, so you can access and return to the information, reference the information in the future. Finally, we're always grateful for feedback. It's really important for the way that we try to improve our training sessions and understand the researchers' needs. So we'd really encourage you to take part in a very short application, a feedback form rather, at the end of the webinar. That'll appear in the chat as well. So we'll appreciate any comments or feedback that you have. In terms of today's schedule, here's a sort of approximate timings. The webinar's split really into 2 big sessions. The first section is with me, and I'll be taking you through a sort of high-level overview of the studies and the value of the studies, as well as taking you through some of the content by subject area. So I'm going to sort of showcase different features of the data. Then I'm going to hand over to Professor Richard Silverwood, who's going to talk about some of the types of analyses that can be done using the data, and then where to go for more information and how to get started with our data. I should say Richard is our chief statistician at CLS, and will be able to answer all of your technical or methodological questions, and I'll do my best to answer those towards the end of the session as well. So please do keep the questions coming in the chat function. Okay, so what are cohort studies? Well, cohort studies are the types of longitudinal studies that follow participants over a period of time. Often, like our cohort studies, they're over many years or even a whole lifetime. Participants share a common characteristic. So, for example, birth cohorts follow an individual born in a particular period, sometimes a day, a week, or a month, or a year. But other cohort studies might share a similar characteristic, like a particular occupation, a particular disease, for example. So these studies follow people throughout their lives, collecting information at particular ages, and they're often referred to as prospective studies. Since everyone shares a birth year, these birth cohort studies means that we are able to look at differences across time and outcome, and those differences are not confounded by generational or historical differences. As a result, birth cohorts are extremely valuable in the social, medical, behavioural sciences because of this continuity, and we can better identify key developmental stages, critical periods, and changes within an individual over the course of their lives. So this slide illustrates that individual over the course of their lives approach, so the life course approach. And they're useful because we can follow the same individuals from birth onwards, or even from pregnancy, so prenatal. So we can observe how early exposures, like in prenatal or in childhood, shape later outcomes. And that makes it ideal as a data set for identifying developmental trajectories, how things change over time. We can look at outcomes at different time points in life, in the life cycle, so post-birth, in childhood, in adolescence, adulthood, midlife, and beyond. And we can use that prospective information to study the risks and exposures earlier in life, as well as to control for a large number of factors in the analysis. And because the data are collected in a sequence, we can see that the factors precede outcomes more accurately. So these prospective studies mean that there's less risk of recall bias or reverse causation. And by measuring exposures before the outcomes occur, these studies can help identify potential causal relationships. So, for example, how prenatal nutrition affects later health or cognitive development. Not only that, because we have information about our cohort members' parents, and in some cases, our cohort members' children, we're able to study the intergenerational transmission of health, wealth, and social advantage or disadvantage. Another feature of the birth cohorts is that we can focus on the period in which participants grew up in. So this slide documents a timeline through the middle where we see the decades covered and maps onto it some of our cohort studies. So there's the 1958, the NCDS study that we'll talk a bit more about in detail today, the BCS 70, Next Steps, who were born in 1990, and the Millennium Cohort Study, who were born just at the turn of the millennium. So it documents the timeline in relation to our studies, as well as some period effects. And period effects are social changes that affect all age groups in a specific time period, like those unique to a birth cohort. So the events or trends that impact everyone living at that particular time. So some examples are here on the slide. There's the Race Relations Act was introduced in 1968. We've got the political trends, Margaret Thatcher features. We've got the introduction of the contraceptive pill. I can't currently find that one now, but that features on the slide. There it is. The IVF, cloning, as well as AI. So each of these period effects will affect everyone simultaneously who are living in that period, but perhaps in different ways. So another example of a period effect is COVID-19. This was an unexpected health and economic shock. Each cohort experienced the pandemic and the subsequent events, but its influence varied depending on their age. So this cohort were in their 60s during the pandemic, this cohort were in their 50s, Next Steps were in their 30s, and the Millennium Cohort Study were in their 20s. So these stages of life meant that they had different impacts on their education, employment, and well-being, for example. And cohort members' individual characteristics and past experiences also shaped how the pandemic affected them. So importantly, the cohorts contained a wealth of information collected before the pandemic, enabling us to study the actual impact of the pandemic on later outcomes without relying on retrospective data. So let me go into more detail about the cohort studies that we'll be focusing on today. These data are a public scientific resource used by thousands of researchers worldwide, resulting in many thousands of publications. They enable the replication and extension of the findings across different countries and time periods, but they're also an important data source for policy evidence. So the first and the oldest of our studies is the NCDS, the National Child Development Study. They were born in a particular week in Great Britain, so England, Scotland and Wales in 1958. They had a starting sample size of around 17,000. The next is the BCS 70, born in Great Britain in 1970 in one particular week, again with a starting sample of 17,000. Next Steps is slightly different. It wasn't a birth cohort study, so we don't have information from early childhood, but instead it started as a school-based study. So it started in year 9 in secondary school in England. And so it's a generation of people born in 1989-90, but we don't have those early childhood years. And lastly, we've got the Millennium Cohort Study, who were born in 2000 to 2002 with a sample size of over 18,000. And I should say this is a UK sample, so it's England, Scotland, Wales and Northern Ireland. And so far I've focused on describing the sort of within individual trajectories. So touching on the intergenerational aspects of developmental trajectories, identifying critical or sensitive periods in development. Here, I just want to highlight the value of thinking about the differences between the cohort groups. So Here you see the nice sequence of between sort of 10 and 20 years between these generations. So it allows us to compare across cohort, so between groups of people born around the same time. And due to their shared long-term exposures of life experience that are unique to their generation, it allows us to think about where we might see differences. So we might consider comparing habits, attitudes, or exposures, like long-term effects of certain diets or lifestyle, and how those differ from one generation compared to another. So, for example, those born in the 1950s might have different long-term health outcomes than those born in the 1980s due to just different life experiences, diet, education levels, even when they're compared at the same age. So this, taking this a bit further in a bit more detail, here are the study timelines for the 4 core cohort studies. This chart shows the years on the y-axis and the age on the x-axis. And this visualization allows you to note where each sweep of data was collected at what age, what time point. So it allows us to think about the spacing of the sweeps between by study and by age and life stage. So you can see for next steps, there's a strong cluster in adolescence into early adulthood, and then there was a gap into sort of early adulthood and mid-adulthood, whereas the other studies have got these earlier life measures documented. What this also allows us to do is read the data points on the vertical, so you could see the timing of the actual data collections within decades. So it allows you to think about reasonable cross-cohort comparisons, so comparing different generations of the same or similar age. So you could see that around age 17, there's quite a strong kind of comparison across all 4 cohort studies. There's reasons why the spacing's different, and that kind of comes down to some funding cycles, fieldwork capacity, and a few other decisions. It's not always straightforward to have the exact same age captured, but this helps us understand the time points and the age points of each sweep. So here's the slide with the actual ages on it. The oldest cohort study of the NCDS has 12 data collections since 1958, and the most latest sweep available is the age 62 to 65 study. BCS has had 11 sweeps of data since 1970, and the most recent sweep is the 51-52 study. Next steps, the most recent sweep is age 32, and that's the 9th overall sweep of the study. And the Millennium cohort study has had 8 sweeps so far. Well, technically 7 sweeps so far. The 8th sweep, the data is being prepared and will be released in early January 2026. So that's technically not available yet, but it's the data has been collected. So part of my job today is to show you the kind of nature and the contents of the studies, and there's a wealth of information in each of the cohort studies, and some are quite sort of specific to the individual studies. So I'm going to talk you through at a very high level in the first instance the types of information collected, and then in the next sort of part of my presentation, I'll take you through some of the scientific themes in a bit more detail. So this slide shows the types of information collected across the life course, so specific to particular life stages. You'll see that there's a vast amount of information collected on the whole, and the content is multidisciplinary, spanning economic, psychological, medical. The columns here show the life stage. So prenatal will capture information about birth weight, labour length. In childhood, we're capturing more types of information about friendships, school subject choices, childcare. And as we move into adolescence, we might change the focus or indeed just add to the focus and include things like risky behaviours, aspiration or peer effects. So these are measures that are specific to the age and stage of the cohort member. The white arrow along the bottom shows the types of repeated measures that are included in the study. And this, depending on the sort of age and stage of the cohort member, this may be of their parents or of themselves. So in childhood, we do capture employment, but obviously that's of the parents rather than the cohort member themselves. And so across the whole life course, we capture measures on health, well-being, employment, education and skills, and family relationships, among many other factors. And that scope of information it allows us to better examine the kind of complex interactions between those biological, psychological, health, or social factors across time. So we might be able to unpick how genetic predispositions might interact with early life environments to influence disease risk, for example. It also, as I've mentioned before, enables researchers to control for many different factors in the childhood development stage. So we might be interested in their home environment when they were a child in relation to their parental class, parental income, parenting style, number of books in the home, or access to computers to better isolate the effect of a variable on an outcome. So, for example, to better isolate the association between parental education and their own educational attainment. So, looking at intergenerational educational mobility. So let me take you through the sort of study-specific features in a bit more of a concrete way. So this is the NCDS data and its structure. So obviously, as our oldest study, it captures the most of the life course from birth up until age 62, 65. So most of the sweeps were in-person interviews. Some came with a nurse's visit who were able to capture some anthropometric measures, so objective health, weight, blood pressure. They took some blood for DNA analysis, as well as to try and understand other health conditions. Where the slide shows height and weight in green, that's just a self-report. But here, where they're in grey, that's where the nurse or there was an objective measure of height and weight. Other things to note then are the early sweeps of the study, the main respondents were the parents, mainly the mother, and the cohort member took part in a sort of minor way for those 7, 11 and age 16 sweeps. So at age 7, they answered a few self-completion questions. At age 11, they included a written response to an essay prompt, imagining how their life would be at age 25. And at age 16, they were involved in cognitive assessments and qualifications and things. And then at age 23 onwards, the parents sort of dropped out of the study and the cohort member became the main reporter. At every age, we ask about qualifications achieved together with their economic activity. So we have quite rich partnership, economic and educational trajectories measured. I think I mentioned this, but this study has quite a strong health focus. It was a perinatal mortality survey initially, so it has really strong health focus early on in particular. And so that was the sort of origin of the study. And since then, it's become much more, well, I think all of them have become much more multidisciplinary, capturing things, in this case, like cognitive assessment, mental health, and as well as the opportunity to link data into the study. So we have a geospatial element where you can link external sources from sort of detailed addresses. So you can find link to things like pollution or green spaces, as well as it being linked to administrative health records, the hospital episode statistics. You will see that the initial number that I started with, that 17,000 participants, just by the nature of longitudinal studies, we do expect them to attrit. So the current sample is around 8,400, but there are lots of ways to handle that missing data approach or attrition. And there's lots of resources that we'll point to later that will help you address this. The next is the BCS 70. It's very similar in structure to the NCDS, although they're just a bit younger. Again, in the early sweeps, it was the parent who was the main respondent, and then it became the cohort member solely at age 26. We have got some school reports of their behaviour, got similar medical focus on the kind of health dimension, as well as similar survey instruments. In addition to the cognitive and mental health, we've also got time use diaries and diet diaries, so what they're eating, and that diet diary was repeated again more recently at age 46. For next steps, this is a slightly different structure. As I mentioned before, it's a school-based sample, so it didn't start from birth, it started at age 14. So we have this cluster of 7 sweeps together annually up till age 20. There was a slight break in the study, and then we follow them again from age 25 onwards, and most recent sweep is age 32. Education was quite a strong focus, so there's lots of measures that capture qualifications, jobs and training, sort of subject choice, in addition to that, but we do still have some health dimensions. We've got health behaviours such as alcohol, drugs, exercise, smoking, and we do have some self-report of weight, birth weight, as well as a weight and height in adulthood, so we can calculate BMI. Similar linkages for the admin and administrative data, so the National Pupil Database and the hospital episode statistics are included, so that's a health and education focus. We also link to vocational administrative data, the individualised learning record, as well as the student loans company data. And we've sought consents for economic data, so from the Department for Work and Pensions, and some of those have already been enacted and are available through the UK LLC. But we're always updating the administrative linked data as a resource. And lastly, the MCS, the youngest of our 4 main cohorts, follows a very similar structure to the others. Parents are the main respondents in the early sweeps, and then the cohort member became their sole main respondent at age 23, the most recent sweep of data due in January for researchers. Similar sorts of things. Hopefully you're getting used to the kind of dimensions, but there's a strong health focus. There's lots of instruments about cognition, mental health included in this data. There's also accelerometry data, so there's an activity monitor at age 7 and again at age 14. There's some time use diaries. I think there are questions on diet, and there's also this genetic data that is included, but actually that's for all of the studies, so I'll mention that in the next slide. These are the types of data linked to the Millennium Cohort study. So again, you can see the kind of wealth of information from either geographic or administrative linked data. And yeah, we currently have a sample size of close to 10,000 at age 23. So some of the kind of cross features of all of the 4 studies, I mentioned genetic data. We have actually that for NCDS, BCS 17, Next Steps, and the Millennium Cohort study. What makes the Millennium study a little bit more distinct is that we have trios of genetic data. So we have the cohort member, the mother, and the father. And all of those studies now have polygenic risk scores that have been deposited at the data archive, so to enable to better isolate, or I guess to sort of remove the association from genetic relationship out of the modelling approach. It's a way to control for the sort of genetic dimension and better isolate the social. Linked admin data I've mentioned, geographical data. We have quite detailed coordinates that data can be linked in, so external sources, like I mentioned, weather, pollution, green spaces, number of fast food outlets in a particular geo area. So there's real interesting opportunities for that data, kind of that data linkage. There's also some harmonized data sets across the cohort. So if you're interested in particular BMI or mental health, you should have a look at those in the UK data archive, and we'll mention how to access that later. And the other feature is sort of not mentioned in much detail on the slide so far, but during the COVID pandemic, we had 3 waves of COVID-19 online surveys across all 4 of our cohort studies, and those were relatively well harmonized to allow for an easy cross-cohort comparison. But it's a sort of snapshot in time to try and understand how they experienced the pandemic and those differences, and lots of research has come out from using that data. There's also lots of new studies that are kind of in the pipeline. Possibly the most exciting of which, but perhaps I shouldn't be so partisan, is that the Generation New Era. This is a new cohort study that's going to start data collection next year with following the lives of 30,000 children and their families. So that's going to add to the sequence of those 4 studies. As part of the preparation for that big new study, we had the early life cohort feasibility study, which was to check that essentially another birth cohort would be feasible in this sort of period of life, and it was deemed it was. And so there's still some data collection that is available for analysis at the UK Data Service, where all of our studies are deposited. So that if you're interested in babies in very young stages, then that's a useful new resource that I think is it's quite new, so perhaps underused at this stage. The Children of the 2020 studies is a DFE commission jointly with CLS, and some of the data are available through a slightly different channel, the ONS Secure Research Service. And lastly, there's the COVID Social Mobility and Opportunity study that follows 13,000 young people who were in secondary school in that particular academic year during the pandemic. So if COVID and education is of interest, then that's a great study to look at. So I'll take a brief breath and then move on to the next section, which is trying to take you through the contents in a bit more detail. It's still sort of I'd say it's not exhaustive, but hopefully it's extensive enough for you to get a sense of what's included across the 4 cohorts. Um, it's basically, this is designed to sort of whet your appetite and give you a sense of the kinds of research questions that you could answer using this data. So you can think about these variables as being the sort of the predictor or the outcome variable. Um, and obviously I'm focusing on these particular subject areas, but there are many, many more. So please put in the chat if you have questions about the contents, but I'm also going to show you how to best search these resources for any particular variables you're interested in yourself. So the the sort of structure of these slides, there's quite a few tables like this, and so the idea is you will get the slides as a sent to you afterwards, so you can use these as a reference. And I'm going to not talk through all of them in lots of detail. I'm going to try and pull out some key bits of information and firstly show you how to read the slides. So the column on the left here captures quite a high level description of the measure. Each of the subsequent columns are the 4 studies, starting from the oldest, the NCDS, to the youngest, the Millennium Cohort study. The figure in the cells denote the age that particular measure was collected. And sometimes the slide will differentiate who the reporter is, so parent or cohort member. So yeah, I realise there's quite a lot to take in. So I'm going to try and zoom in on particular aspects so that it'll help sort of situate the information a bit more. But I appreciate it's something that you're likely to need to come back to later. So just picking out a few quite interesting ones. Self-assessed general health is a useful summary variable of health, captured quite well across the 4 studies. Less so in next steps, but it's captured well in the other 3 repeatedly. We've got the linked hospital episode statistics data, the anthropometry, so capturing objective measures of blood pressure, body fat, grip strength, vision, motor skills, for example, as well as height and weight. There's measures of drug and alcohol consumption, which might be seen as a risk factor, and also lots of information on smoking and long-standing health conditions. So you can think about using this data to track changes in perceived health over time using the self-assessed general health. It might be analysis of incidence and prevalence of hospitalizations using the linked data, or identifying how activity or sleep patterns are associated with obesity, for example. So you can start to think about them as exposure or outcome variables. These are the sort of COVID sweep specific information. So again, it's just a kind of high level snapshot. We have antibody tests for the 4 cohort studies. We've got self-reported measures of long COVID symptoms and testing, as well as disruption to medical appointments. And so these are captured at the same time over 3 sweeps, or 3 waves in this case, during the pandemic. So if you're interested in health, physical health or biomedical science, then I would just point you to one of it's a sort of joint collaboration with Closer, who looks after all of the UK longitudinal studies. And this features information about the nature in a lot more detail about the biomedical science perspective. And so you can access that both on our YouTube channel, but also through the link on these slides that you'll get afterwards. Moving now to mental health and well-being. So there's lots of different ways to measure mental health. There's sort of symptom-driven scales, screening tools for diagnoses, and they vary by cohort and over time, partly because the director of the studies were selecting the best scientific knowledge at the time and the age appropriateness of the measure and the expediency, so this how efficiently it could be completed within the sweep. Study sweep. So I'm not going to go through all of these scales, but I will just focus on the malaise scale to show you the types of information collected. Each of these scales come with a validation and associated papers that you can cite in detail. So they are well sort of tested. And so the malaise inventory is actually, this was a 9-item version that was devised by the late CLS professor and former director John Binner. It was basically, he reduced it down from the original 24-item scale. that was developed from Cornell Medical Index Health Questionnaire created by Michael Rutter. So this 9-item malaise inventory has been found to provide accurate and consistent measures of psychological distress, both within and between generations. So it's a validated scale. And basically in the questionnaire, you would score 1 point for every yes and 0 for every no. And if you have 4 or more on these types of symptoms that are associated with depression, then you would be considered to have be at risk of poor mental health. So the types of questions: feeling tired, miserable or depressed, irritated or jittery. So you can get the sense of the kind of contents. And each of these other scales have similar but different items and wording. And again, they're designed. They were selected based on the most age appropriateness and the sort of the prevailing knowledge at the time. So malaise is used for the older 2 cohort studies, BCS and NCDS, and pretty well measured across adolescence and adulthood. So you can see that it would be a useful thing to track fluctuations in mental health or mental health trajectories over time. The other measures here, Millennium to make use of the SDQ, so a similar thing where you can measure change over time. The additional value of this type of measure is that it's both the teacher and the parent, as well as self-report. So you might be able to sort of do a bit of a measurement study, so comparing teacher-reported versus parent and child-reported symptoms of mental health to understand context-specific behaviour, or look at how one tool maps onto another. So there's lots of opportunities for exploring mental health in the studies. These are quite recent measures that have been included in age 32 next steps, age 23 Millennium cohort study, and all of the COVID-19 specific sweeps. So these are 2-item responses that capture depression and anxiety, and that there's so opportunities for some cross-cohort analyses and within individual analyses using those for next steps and the Millennium in particular. Again, we've got a standing resource on our YouTube channel for the mental health in the 4 British cohort studies. So there's 45 more minutes of talking about mental health on there, if that's a particular interest to you. Obviously, I'm just taking you through quite a high level description of things right now. Moving now to well-being. So we've covered mental health. Distinct from mental health, or maybe an aspect of, is measures of well-being. These are more considered to be like a sort of cognitive appraisal of satisfaction or health. So lots of different measures here, in particular life satisfaction, which is measured either by the parent or the cohort member themselves across the course of their lives. And with these data, you might look at how, so the other things to mention rather is self-efficacy, sort of belief that they're capable individuals. Loneliness are measured also in the data using some of the UCLA measures. And so with these type of data, you might look at how predictors of life satisfaction might differ with predictors of mental health, or how loneliness is associated with other health outcomes. Moving now to family and relationships. We measure this at every sweep. Essentially, we're interested in who is in the household or who's in the family, capturing family change in quite a lot of detail, so those trajectories. So these are the kinds of things we capture: age, biological step, adoptive parents, so the relationship to the cohort member, fertility intentions, pregnancy histories, as well as quite detailed partnership formation. So these types of questions you can answer are things like whether birth order influences cognitive development or educational pathways, what is the impact of household instability, so the frequency of changes of family members in the household might be associated with differential child behaviour or well-being. There's quite a well-established association between parental age at birth and child's educational attainment, or indeed child's own fertility. So there's an intergenerational aspect of it too. And also you could answer by looking at fertility intentions, whether different fertility intentions are associated with more parental investment or better childhood well-being. So again, just pointing you to another resource on the YouTube channel. This is another webinar in 3 parts. So there's around 45 minutes, 50 minutes on families and relationships in a lot more detail than I can go into today. got two more sections, so just very briefly, education and ability. So education measures are captured objectively through those data linkages I mentioned for Millennium and the Next Steps cohort studies. For NCDS and BCS, there's, I think, school reports, but there's lots of detail on the subjects that they've chosen. And we update the data based on their most recent changes. So if we ask every time we ask whether they've done any new qualifications since we last spoke to them. So we've got an up-to-date measure of highest qualification, as well as things like university type, subject studied, or degree grade. So with these data, you can document educational trajectories, you can answer questions about intergenerational parental education, or look at differential labour market returns by degree subject, for example. We do measure some of those standardized tools I mentioned, the instruments to capture cognitive ability, so distinct from qualifications. These are well-validated, renowned tests, such as the British Ability Skills Test or the Bracken School Readiness Test. And they're measured quite well, I think, over the three sort of more health-focused data sets, and less well in Next Steps, but we've captured short-term working memory in Next Steps at age 32. It's a really interesting area of work. Often the skills that you require to access these types of tests are pulled from others. So even if you're doing a mathematical or a numerical test, you will still need to have good verbal skills or literacy skills in order to access and follow the instructions. But here the one I've highlighted is the verbal skills, which has several separate tests measuring reading, language comprehension, spelling, and word definitions. So with these data, you can chart the cognitive trajectories from childhood to adulthood. You can measure growth rates in verbal and non-verbal memory or processing speed domains, identify critical periods where cognitive inequalities widen or narrow, for example. So lots of opportunities for research on that topic. So lastly, we're on to occupation earnings and income. Earnings where money is obtained from labour or services, income where it's both work and through investments or benefits, and those are well captured across the life course, partly reported by the parents in sort of childhood and adolescence, and then self-report as they age. We also have measures of wealth, an accumulation of valuable economic resources, so housing wealth, the combination of savings, debt, and pensions. And so the types of research questions that could be answered here are those life course socioeconomic trajectories. So how do earning trajectories from early adulthood predict wealth accumulation later in life? To what extent does parental income in early childhood shape adulthood earnings and wealth? And how does family income at different developmental stages relate to cognitive outcomes, for example? So you can see that sort of exposure and outcome across the different domains that I'm talking about today. Okay, so how to search the resources yourself. This is very brief, and then I'll hand over to Richard. So first, I'd say go to our website. There's a section called Exploring Our Data, on which there are links here to the data dictionaries. I personally, this is my favorite way of searching. So the Excel spreadsheets have sweep, year, age, data name, topic, subtopic, variable name, and as well as the actual questionnaire wording. And as with Excel, you can filter and search through particular things. So you might be interested in a particular age, 16, for example, within a particular study, and that you're interested in alcohol consumption, and it will filter through all of the data in relation to that topic. So that's the sort of back end version that you kind of a bit more manual. There is a sort of a more user-friendly approach, and this is currently being beta tested, so it's going to be in constant development for a little while, and we're going to be releasing improved versions as we go. But the slide is actually linked so that you can search for it yourself, and here you can just put in the study or the sweep that you're interested in, the category, the research topic, or the variable name if you know it. So it's a way to filter through essentially what the contents of that Excel spreadsheet were. Closer Discovery is another search tool. So if you're interested in the studies, the longitudinal studies outside of our 4 studies that we're talking about today, So you can use closer discovery. So it has the whole of the UK longitudinal studies in it. And here I've just searched for life satisfaction in the British cohort study, and it tells me the data set it's in, the age it's captured, and the exact question wording. And lastly, you might just search the actual questionnaires that are available on the website or at the UKDS. You can download the actual data sets and search the variables, or look through the published papers, most of which can be found on the CLS bibliography. So you can look at the variable description that researchers have used before you, and again, their modelling approach and things as well. So sorry, that was brief but hopefully informative. I'm going to hand over to Richard now, who'll take you through some of the research analysis opportunities using this data.
Right, thank you all for joining us today. Thank you, Morag. So I'm going to talk next about some examples of the types of analyses that you can undertake using the data that Morag's now introduced. So obviously you've seen the incredibly rich data that we have across the cohort studies. I'm sure you can already envisage loads of fun analyses of different types that you might want to undertake. So I'm not looking to give some exhaustive view of the possible types of analyses, really here just thinking about a small number of analyses that I guess leverage particular features that we have in the CLS cohort studies. And each time I'll do so by introducing them using a published analysis, but I won't go into the details of the kind of the specifics of the analyses, because that's not really the message that I'm trying to get across. So there's lots of more complicated analyses you'll do, but I'll start off with some very simple, more descriptive analyses, and then build on that. And as I say, look at some of the particular features of the cohort studies that can be utilized. So I think I've said this already. Complex analyses are possible, but let's start with some very simple analyses. So first of all, an example that's taken from this briefing document that was published recently, looking at fertility intentions and postponed parenthood using the most recent data from Next Steps at age 32. So initially, there's some analyses using the responses the cohort members gave to this question. Which of these statements best describes the way you feel about having children or having more children as appropriate? And you can see there's a range of different responses, from definitely liking to have children or more children and actively trying at present, through to definitely not wanting to have any further children. So you can do some simple descriptive analyses using these responses. The purple bar here gives the distribution of the overall responses, and the black and the sort of light pinky color bars give the responses stratified by existing parents and those who don't currently have children, respectively. So you can see here that there's some differences, as you may expect. Amongst parents, the proportion who definitely would definitely not like to have more children is higher than amongst those who are not parents at age 32. The next set of analyses in this briefing utilize the responses to this question. So which of the following best describes the main reason you are not currently trying to have children or more children? And this was only asked to those who had given the appropriate response to that first question. So you can see here there's a number of different potential responses that I won't read out, and the respondents could tick which of these they felt was the most appropriate response for them. So here again, we can see the distribution for the reasons for not trying to have children or more children right now. You can see the most prevalent response is that cohort members didn't feel ready to have children or more children at that point. So we can go on and we can drill down into this a bit more. We could split it by any number of different socio-demographic characteristics that we've captured data on. But here you can see some results split by whether the cohort members had a degree or didn't have a degree at age 32. We can see that there's a slightly different distribution here, and a relatively higher prevalence of those who do have a degree said that they did not feel ready to have children or more children relative to those with no degree. And you can see here, once you're starting to look at the kind of the differences in some variable dependent on the value of another, you might want to go on and fit sort of a regression model or estimate a risk ratio or an odds ratio or something here. And so that's what we'll go on to thinking about next. But first of all, the key message here is that we've got literally thousands of interesting variables collected across the cohorts. So there's a great opportunity for this sort of simple or more descriptive analysis. And indeed, even if you're going on to do more complex analyses, I think inevitably you'll start off with some more simple descriptive analyses anyway. So if you're going on from there and building up, perhaps fitting some sort of regression models, then one thing, if you want your estimates to have any sort of causal interpretation that you'll need to worry about is the potential for confounding. So I'm sure many people understand what I mean by confounding, but just very briefly to ensure that we're on the same page, a confound is a variable that causes some non-causal or spurious association between your exposure or your independent variable and your outcome or your dependent variable. So a toy example here, we're interested in the effect of some exposure variable X on some outcome Y. If we observe in our data that there's a statistical association between X and Y, are we certain this is causal? Or perhaps could there be this third variable Z, which is a common cause of X and Y, and would therefore cause some correlation or some association between X and Y. If we think that variable Z might exist, then we would want to control for it in our analyses. Or for a more concrete example here, we're interested in the potential effect of coffee on cancer. We would want to think about whether there's any potential for confounding. So one obvious factor here we might consider is smoking. If in the population, people who drink coffee are more likely to smoke, and smoking itself causes cancer, then smoking could be an important confounding variable that we would want to control for in some way in our analysis. And thankfully, we've got very rich data on our cohort members going back many years or decades, provides a great opportunity for confounder control. And this is one of the main reasons that people like to use this sort of long-running cohort data. So I'll take some example analyses here from this paper looking at the association between early life mental health with biomarkers in midlife and premature mortality, though I'll only focus on the biomarker analyses. So the question here is whether there's some causal effect of early life mental health on biomarkers in midlife. So if we're interested in this, what sort of confounders do we think there might be that we should account for in our analyses? Oh, sorry. First of all, just to say how these were captured in the data. So early life mental health using the Rutter scale that I think Morag mentioned already, particularly in terms of conduct problems and affective symptoms, and a number of different biomarkers that were captured at age 44 to 45 from samples that were taken at that sweep of data collection. And sorry, back to potential confounders. So there could be a number of different types of confounders we could be interested in, perhaps different characteristics of birth, parental characteristics, early life socioeconomic position, early life cohort member characteristics. And we observe lots of different variables relating to all of these different kind of constructs. So I won't read through all of the different variables, but we observe lots of information about the birth or around the birth, such as birth weight, maternal smoking during pregnancy, lots of information on parental characteristics, such as employment and various aspects of home life. Again, lots of variables measuring early life socioeconomic position across a variety of different dimensions. Early life cohort member characteristics. Again, many of these have been mentioned already, cognitive ability, etc. And in this analysis, in this published paper, all of these variables were controlled for in the analysis. And so in that way, the authors hope that they can control this potential for confounding as much as possible. Though I think in that paper, they did go further and conduct some sensitivity analyses considering the potential for additional unmeasured confounding. And I think an important thing to note here is in this type of cohort study, because we're following individuals up from birth, all of these variables are observed prospectively. If we were observing them only retrospectively, as we would have to do in a cross-sectional study, then there's a much greater potential for misreporting and potential bias due to the confounders not being well captured. So this is a clear strength of cohort studies. Excuse me. So here the key message is really that we've got very rich data collected prospectively over many years or decades, and we've got a great opportunity for this sort of really careful confounder control. Another feature that I think has already been mentioned already is repeated measures. So in each of the cohort studies, we're interested in topics that we managed to observe fairly consistently over the life course, with repeated measures of the same or a very similar measurement or construct. So some examples of this would be sort of physical, anthropometric measures, measures of health and mental health, and a variety of different socio-demographic characteristics across the life course. And having these repeated measures of the same or very similar measurements or constructs allows us to characterize the changes or the trajectories of these features of people's lives over time. So an example here using data from, I think, NCDS and BCS 70, looking at patterns of eczema activity from birth through midlife. And so here the authors aim to identify different subtypes of eczema based on patterns of disease activity. So in these two cohorts, there were repeated measures, both self-reported and parent-reported, of period prevalence of eczema, using standardized questions at different points in their lives. So these data, these reported data points, were used to construct subtypes of eczema, which I'll show in the next slide, which may be of interest in and of themselves, but also the authors here went further and used these identified subtypes both as outcomes and as exposures in subsequent analyses. So it's just to emphasize that the trajectories themselves might not be the endpoint, it might be part of a broader analysis. So here it shows the results of the identified eczema subtypes, with NCDS on the left-hand side, BCS 70 on the right-hand side. On each subplot, it's got age on the x-axis and the predicted probability of having eczema on the y-axis. So you can see that in each of those studies, 4 different subtypes were identified, which I won't discuss the particular nuance of those or the methods that have been employed. The key is just we've got these repeated measures and in a broad range of different dimensions of people's lives, we can use to characterize changes or trajectories over time. The final thing that I wanted to look at is the potential for cross-cohort analysis. So this has already been mentioned a bit as well. So within these birth cohorts that we have, because they're very narrowly defined in terms of when individuals were born, we can address some very interesting hypotheses about what's happening with age, what's happening with time. But if we can combine information across different cohorts, across multiple cohorts, then we can extend the analyses that we can do, extend the hypotheses that we can address. Then we can start to think about how things change over time or between cohorts. So if we're trying to undertake this sort of endeavor, combining data across different cohorts, ideally we would want to analyze identical measures across cohorts. Sometimes this is possible when we're combining data from cohorts that each of which we're running ourselves, and we can prospectively try and ensure that the measures that we use are as similar as possible across cohorts. If this isn't the case, and this, if we're using older data or using data from different places, this may well be the case, we need to consider how measures can be best harmonized. So how can we perform some manipulation of the data in advance to make these measurements as comparable as possible between the cohorts? So again, I'm going to discuss some points around this using a published paper. This one looking at socioeconomic inequalities in childhood and adolescent BMI, weight and height using 4 of the British birth cohort studies. And here I'll just mention the results relating to BMI. So the authors here were using data from 3 of the CLS cohorts that have been described already, NCDS, BCS 70, and MCS, as well as data from the National Survey of Health and Development, is another birth cohort born in 1946, run by another team also based at UCL. And here there was 2 or 3 data points in terms of BMI observations within each of these different cohorts. Socioeconomic position was indicated by the father's occupational social class, which was reported at age 10 or 11 in each of these cohorts. And the authors examined the associations between childhood socioeconomic position and BMI to assess these socioeconomic inequalities. So I'm just going to show this one plot of the results. There's a lot more results in the paper. This looks like a very simple plot, but there's actually a lot going on here. So we've got age on the x-axis, BMI on the y-axis. Each of the different colors relates to one of the different studies. The dashed lines and the solid lines relate to the lowest and highest social class groups, respectively. So from this plot, we can still make within cohort comparisons. So if we look at a single line relating to a single cohort, we can see whether BMI changed within that cohort and within that social class group with age. We can make comparisons between the different social class groups in the same cohort by comparing the dashed line and the solid line within the same cohort. So if we do that at one point in time, we can see whether there were differences in BMI or social inequalities in BMI at that age. If we compare the slopes of them, so the same color, dashed versus solid, we can see whether that was increasing or decreasing over time within the same cohort. But we can also now make between cohort comparisons. So if we're comparing lines of the same type, so either dashed or solid across different colors, so across different cohorts, we can see whether there were between cohort differences either at a certain age or over time in terms of BMI. And again, if we compare the differences between the dashed and the solid lines within cohort, but make that comparison across cohorts, if you see what I mean, then we can see whether as time progresses, there's been differences in changes in inequality in BMI over time. So there's lots of very nuanced interpretations that we can make here. It's a very powerful type of analysis. This allowed the authors to draw conclusions such as this, that there was little inequality in childhood BMI in the earlier cohorts, inequalities present in the most recent cohort, and the inequalities were widening from childhood to adolescence in all the cohorts except the oldest one. So again, key message here, using this sort of analysis, if we're combining data across multiple cohorts, it allows us to extend our hypotheses and think about how things change over time or between cohorts, as well as just within a single cohort. If you're interested in this type of analysis, we have a paper a couple of years ago now that's looking at some of the issues around this that you would want to consider if you're undertaking this type of analysis. So I've only looked at a few different types of analysis. I've only, given a single example for each one. There's thousands of papers that have used the data from the CLS cohorts. So if you're interested in finding out a bit more about the different types of analyses that have been undertaken previously, then there's a CLS bibliography that's got a comprehensive catalogue of all of those different papers, and that's a good place to start. Okay, I'll pause for 1 second and I'll move on to the next section. Okay, right, so getting started with the data. So here we're going to think a little bit about how you would, in practice, go about accessing the data from the CLS cohort studies, some additional resources that there are around the data themselves. Some of these have been mentioned already. Then think about a few of the characteristics of the data sets and the studies that you need to be aware of once you're embarking on analyses. And finishing off with several slides on where you can go for more information, many of which have already been mentioned, but just to emphasize some of those again. So I've given this statement its own slide and big font because it's important. So researchers in the UK and around the world can access data from our cohort studies free of charge. So this is very important, and this is a major role that we serve, is to create these really important resources for the research community that are available for everyone to use. In practice, how do you go about doing that? So data from the CLS cohort studies are all available, almost all from different data repositories. The vast majority of our data are available from the UK Data Service, which is a digital repository for data across the social and health sciences in the UK, available at this URL here. So the way that our data are available on the UK Data Service depends on the categorization of the data. So there's 3 categories that are essentially low, medium, and high levels of disclosure risk and sensitivity. So starting on the left here, the lowest risk are what we call safeguarded data. And these are available from UKDS under what's called the end user license. So as I say, these are the lowest level of risk. The vast majority of our data are available under this category, so under this license. Very straightforward, almost instant access to the data from UKDS, and we'll go through in the next few slides to look at step-by-step process of how you would do this. The application is approved directly by UKDS, and then you would download the data from UKDS. They're available to use on your machine locally. The next category are special safeguarded data, which are available from UKDS under special license. And these are moderately sensitive or moderately disclosive data. So these have an additional step where the application must be additionally approved by us at CLS, so not quite instant, but the approach by which you access the data is exactly the same. They're downloaded directly from UKDS. So again, still very straightforward. And then the final category of data, referred to as control data, which are available from UKDS under a secure access license, are the most sensitive or potentially disclosive data. So these include the sort of linked health data and other very fine-grained information, including some geographic, geocoded data that we allow access to. So here the application again has to be approved by CLS, but the major difference is that you're not able to download these data and use them locally on your machine. You have to access them via a trusted research environment, which for the UK Data Service is called the Secure Lab. So it's a ring-fenced environment where you can't take things out unless they've been thoroughly checked. And in order to use that, you have to attend some specialized training. So if you're wanting to access this sort of data, then it can potentially, particularly the first time you try to access them, it can take quite a while, potentially a few months. So if you're looking to quickly conduct a research project, for example, if it's part of an undergraduate or PGT dissertation, then there's certainly risks involved on the timescale for accessing data. If you're interested in this, more information on the CLS website at the link at the bottom of the slide there. Okay, so I'm going to go through now and just go through how you would go about accessing some of this lower risk data under an end user license from UKDS. So on the UKDS website here, I think it's best to first of all log in before you do anything else. Login button on the top right. And as it says here, if you're at a UK college or university, it's likely that you can sign in using your username or password from your college or university. So for many people, that's the way that you do it. You select your organization in the box here, and I think this includes government departments and other organizations too, and that will allow you to log in a straightforward way. If your organization's not there, then you need to add in a few details as part of the registration process, but it's still relatively straightforward. When you're trying to locate data sets on the UKDS, there's a number of different ways that you can do so. If you know exactly the data set that you're after, so a certain cohort, a certain sweep of data collection, a certain type of data, then there's a search box. You can try and search directly for it. You might be able to access it that way. All of the data sets, all of the deposits relating to an individual CLS cohort study are also tagged so that you can locate them all grouped together. So these links here are direct links that take you into a full list of deposited data sets for each of the CLS cohort studies. So if you were to click on the link here for the Millennium Cohort Study, it would take you to a page that looks like this. So this, if you scroll down, gives you an overview of the Millennium Cohort Study. And there's various bits of information here, but let's focus on accessing data for the moment. So if we were to do so, we would click on Access Data. And that would take us to a page that looks like this. So this gives us a list of all the MCS data sets, all the deposits that are available within UKDS. So we can see here, there's a few at the top from the early sweeps of data collection. So these ones are available under a standard end user license. And towards the bottom, there's a few more. So one with linked educational administrative data in Scotland towards the bottom, and then the three at the bottom with different geographical identifiers, and those ones are only available under secure access. So all the data sets are listed. You can click on them and find out more information. But let's go for a standard end user license one, because that's the most straightforward. So let's say we want to access this data set, MCS age 3 sweep 2 from 2004. If we click on the link there, it takes us onto the page for this data set. Again, lots of information there on this first tab with details. If we scroll down, the next tab will give us a list of the documentation that's available with this data deposit, including the user guide, the questionnaire, etc. We'll discuss some of these later on. You can access those directly here without having to download the data, but all of that documentation will come as part of your download once you do download the data anyway. So if we want to access these data, we click the button here in the bottom left to add this data set to our account. So in UKDS, there's an additional stage here before you can download the data. Before you can download any data set, it has to be assigned to a project. So a project is essentially just a space that you have dedicated to a specific research project that all of your data sets live within. So if we were trying to download this data set here, and we want to add it to a project first, if we were trying to add multiple data sets at the same time, they'd all be listed here. But here we're only trying to add this one, put a tick in the box, click Add to Project. It will take us here. So if we already have an existing project, we can select it from the drop-down menu, add this data set in here and carry on. Otherwise, we need to create a new project, but that's very straightforward. You just need to give it a title and a bit of text as a kind of overview of what you will be doing as part of your project. Once we've added it to the project, we can go to the project page here. So this is an example that already had 2 data sets within it. We've just added this additional one from MCS age 3. We can see that this yellow request access box is telling us that we have some additional actions to do. So if we click on the actions drop down here, it will tell us what we need to do. The first thing is that we need to ensure that we've accepted the standard end user license. So this is kind of a user level agreement. So once you've accepted that once, then this should show as being accepted forevermore, I think. And then there may be some additional conditions of use that relate to this specific download. So here, of course, you need to carefully read these and ensure that you're happy to agree with these conditions. When we then go back to the project page, we should see that it's magically gone green to show that we've done everything that we need to do. We can select it and click download, and then we go to the download page. From the download page, we can select our file format of choice, click download selected, and that's it. We will have the data available to analyze locally. So I've probably made it look as complicated as I possibly could, but once you get used to the process, it's relatively simple. You locate your data, add it to your account, add it to your project, ensure that you've agreed to everything you need to agree to, and it's available for download. So some additional resources here in terms of the data sets themselves. I'll come back to more general resources later on. So these are some of the documents that I've already mentioned and that Morag's mentioned already. So all of these, with the exception of cohort profiles at the end, are available on UKDS and will come as part of your download when you download the data. User guides give detailed information about everything to do with the deposit. We always develop a detailed user guide for each deposit, so there'll always be one, and this should be your primary kind of point of contact with the data set. So it gives an overview of all of the measures that have been used, the fieldwork processes, the levels of response. If we've got weights that have been derived, it will describe how that's been done. The questionnaires that have been used to collect the data will be there, which is useful. Often you'll need to know the exact question wording for different purposes. There'll be other data documentation, particularly in terms of derived variables and the way that the data have been processed and recorded. Technical reports, which might come from outside agencies, will give even more granular information on different parts of the fieldwork process, the coding. And then outside of that, there's published cohort profiles for each of the 4 CLS cohorts. So they're listed here. So these, give by nature, they have to be quite short, so they give more of an overview of each of the studies. But these are the primary reference that you would want to cite in work if you're using the data sets. Then they do give a good overview of the processes behind the selection, the data collection, and the data that's available in each of the cohorts. Okay, a little bit on the data sets as you might find them and some of the key identifiers. So here, if you were to look at all of the data sets that are available for Next Steps Sweep 8, so in 2015, you might find that you've got a download basket that looks something like this. So we can see that there's several different data sets relating to different aspects of the data collection. Some of them will be the survey data with some processing, but more or less as it was collected. Some of them will have had some derivation. And the one at the bottom is specifically a data set of the derived variables, which will come with its own documentation. In the final column there, you can see that I've noted the structure of these. So the data sets will come in different structures. So a flat structure just means that there's one row of data per cohort member in this case. So that will be the majority of the time. The standard survey data will come like that. But there's also data sets that are hierarchical, so potentially with more than one row of data per cohort member. So anything where there's potentially more than one entry, so anything that's like a relationship history, employment history, time use diaries, that sort of thing may come in a hierarchical file, and you just need to be aware of that. Cohort members have their own consistent identifiers, which are used across the data sets within the same sweep and across sweeps. So in NCDS, BCS 17 Next Steps, there's just one identifier for each cohort member. In MCS, it's slightly more complicated, as the standard identifier, MCS ID, identifies the families, and there's additional identifiers that are required to identify the cohort members within the family and anyone else, the other family members. So these IDs, because they're used consistently across the data sets, these can be used to combine or merge data sets to ensure that you're connecting together data for the same cohort member. So there's some supporting documentation around how to do that. Particularly this data handling guide for MCS is handy because of the additional complexity around the IDs in MCS. A bit now on the design of the studies and the survey variables. So sorry, this is a bit of a text-heavy table, but I think some of this information has already been given by Morag. So in NCDS and BCS 70, the design of the study was just to capture all births in 1 week in Great Britain. So there's no what we would call complex survey design. In Next Steps and MCS, there is this sort of complex survey design. In Next Steps, first of all, schools were sampled, and then within those selected schools, pupils were selected within those. And there's also some oversampling, so more deprived schools or pupils from minority ethnic groups were relatively more oversampled. And so what that means is, if you were to just analyze the data as is without accounting for this, particularly this oversampling, then you wouldn't have a representative sample. So you need to account for this complex survey structure in your analyses. And you can see in the final column there, I've listed the survey variables that you would want to account for in your analyses. So the primary sampling unit here is the schools, the strata, the different kind of population subgroups that were differentially sampled, and the design weight will re-weight individuals back to the population. And so there's information in each of the relevant user guides about how you would do that. MCS also has complex survey structure as described there. And again, there's documentation to help you account for that appropriately within your analyses. I've realized that I'm likely to go over, so quickly going through the next couple of sections on non-response and attrition, first of all. So there's a couple of different types of non-response that you might want to consider when you're using these data. The first is sweep level non-response. So this is cohort members who who we know are in the study, but don't respond at all at a particular sweep. And that might be because we've lost contact with them. There might be a permanent refusal. It might just be because they've refused at this particular sweep to participate. This is very common in longitudinal studies. So we can see in this figure at the bottom that our number of respondents for NCDS at birth in 1958 was 17,000 and something, and by age 55 had dropped down to about 9,000. So we can see that we've lost almost half by that point, which seems like a large number to drop, but I mean, the fact that 55 years later we're still able to collect data on such a high proportion of the initial cohort, I think is something of a miracle and testament to the hard work of lots of different people in different teams at CLS. The other type of non-response that you get is item non-response. So that's cohort members who have responded at a particular sweep, but have just chosen not to, or have perhaps by accident not answered a specific question or given data for some other item. This tends to be less of an issue in the cohorts. Sweep non-response is a bigger issue. And when you've got this sort of non-response, then you have essentially a problem of missing data. And when you have missing data, it reduces the analysis sample size that you have, so there's lower statistical power. But a more important risk here is the potential for issues of representativeness. So we know from work in our cohorts and elsewhere over many decades that respondents to this type of survey will often systematically differ from the non-respondents in a number of potentially important ways. So if you were to conduct what might be called a complete case analysis, so analyzing just those cohort members with observed data, then that might lead to bias. So in your analysis sample, you're likely to have individuals with higher levels of education, lower levels of deprivation, better health. We know they're more likely to respond. So the conclusions that you draw might not generalize to the entire population that you're interested in. So there's different approaches that you can use to try and address this problem. One is the use of non-response weights, which generally try to re-weight your analysis sample to be representative of your original sample and therefore your target population. So with some of our data sets, well, all of the sweeps of MCS and Next Steps, the most recent sweeps of NCDS and BCS 70, we've deposited kind of sweep-specific non-response weights that can be used to re-weight the respondents at each of those sweeps. So in the relevant user guides, there's full details of how we've derived those and how they can be applied. You can also go further and use and derive your own analysis-specific weights using the sorts of approaches that we describe in the user guides. There's other methods that we discussed that I've not got time to go into now, but if you're interested, there's a lot more information. There's a handling missing data page within the CLS website. From there, you can access this handling missing data user guide, which is, I think, really the main source of information on this with lots of detail. And there's also one or possibly more videos of webinars that we've run on handling missing data in the cohorts. Where to go for more information? Very quickly, so we've still got time for a Q&A. Some of these have been mentioned already, so I'm repeating things that have already been said. CLS website has study-specific pages which have lots of information, including dedicated introductory videos for each study. I've already talked a bit about user guides, technical reports and questionnaires available from either the CLS website. UKDS will come for free with your downloaded data. On the CLS website, there's also our training and support page where you can find information on upcoming training events and also watch recordings of previous webinars that we've run and sign up to ensure that you're alerted when new training opportunities become available. But a kind of fuller repository of the videos from our webinars is available from the CLS YouTube channel here. And as well as the study specific webinar recordings that I've already mentioned. There's more specific ones on latest data releases, specifics about approaches for methods and data handling by different scientific theme and different types of specialist data. Closer have an emphasis on kind of accessing and using data from longitudinal studies in the UK. They have a learning hub, which has different learning modules that you might be interested in, and you can see on the screen there. National Centre for Research Methods run training for research methods, as the name suggests. And so they have lots of relevant different in-person webinar and recorded training opportunities there. The UKDS that we've already met also have a learning hub with lots of different training across the different types of data that they have there. And I will wrap up there. So we have had an introduction to the birth cohorts. Morag's given lots of information on the content by different subject areas. I've given a brief overview of some of the types of analysis you might want to undertake, as well as how to access the data and where to go for more information. Thank you.